Now, I am sure that you will be going through some Punnett squares, and that will be something that you will see on an exam. And there are slides within this PowerPoint presentation that walk you through the steps as to how to complete those Punnett squares. Now, I want to talk about several diseases and how they work. And all of these are going to be about diseases that are linked to one particular gene. And I want to make it clear that not every phenotype is dictated by one gene. So what do I mean by that? Your skin color, for instance, there are 10 different genes with different alleles in those genes that contribute to your skin color. So again, when I say that Mendel was lucky, he picked the flower color or the pea color or whatever he picked, and these were single gene traits. And so let's look at a few other single gene traits. Cystic fibrosis is one of them, where this is just showing us a pedigree of males and females. This is a recessive trait. What that means is when you have the lowercase letter that in combination with the uppercase letter, you will not have that disease because the dominant good version of the gene will do its job, and it'll take care of the job for both of the alleles. Okay, so that's a recessive trait. Now, the explanation for that is that a single copy of the dominant allele makes enough protein to give you the normal phenotype. Okay, so let's take a look at some other recessive human traits. There is albinism, phenylketonuria, which we talked about before. This is a boy in the plastic bubble here, old John Travolta movie, and this is a trait that is a recessive trait. Sickle cell anemia. This is xeroderma pigmentosum. This, all of these, in order to have this disease, you must have 2 recessive genes. Otherwise, you would just be a carrier of the trait, or maybe not a carrier at all. We do have a student in our class who introduced me to a disease syndrome called MCAD, and this is also an example of a recessive trait, and I will talk to you more about that later. Now, let's move on to, we know disease genes can be recessive or dominant. We know that the examples that I just showed you were recessive. But here is the plot twist. What if the disease gene is dominant? And that can happen as well. And I will show you an example of that. We have here Huntington disease. And the pedigree looks different if you want to study pedigrees. And the Huntington disease, the dominant trait or the dominant allele is the disease gene. It causes a neurological breakdown, and it is something that typically would impact you in the later part of your life, 50s, 60s, 70s. And because it is causing misfolded proteins that don't allow your neurons to communicate, one version of the bad gene is enough to make you sick. Okay, second plot twist. Disease genes can be autosomal or X-linked. Let me get some of that stuff out of the way here. So everything that I've taught you so far has been autosomal, meaning chromosomes 1 through 22. But you can have genes and traits come from genes on the X chromosome. And an example of those are color blindness or a particular type of hemophilia called hemophilia A. Because men don't have 2 X chromosomes, they only have 1, we call men hemizygous for X-linked genes. So this plays out in a much different way. Let's look at the example here. This is a Punnett square, right? We have the female gametes here and the male gametes here. Female, of course, are giving an X chromosome. That's all they have to give. And the male is giving either an X or a Y chromosome in the sperm. Now you can see here that this female happens to have one good copy of the gene, and this is a bad copy of the gene. Hemophilia is a disease where you don't clot your blood, so you would bleed to death. The male in this case can only give, he's giving a good version of his hemoglobin on this X chromosome, and the Y chromosome doesn't have that allele. And now what we can see is that when we have the children, right, we have the two females here, we have two males here. This female is unaffected, right? She doesn't have the disease. And actually this female is unaffected as well, but we call her a carrier. So she's got the good version of the gene that's doing enough for both, even though she has a bad copy over here. So she's not going to have hemophilia, but she carries that gene. And then for the men, you can see this is an unaffected or a healthy male, whereas this male has hemophilia. So with X-linked traits, men have a much greater chance of having these diseases because they are hemizygous. And to just bring a little bit of history in, we believe that this broken gene for hemophilia came from Queen Victoria. She had many children, many grandchildren, and if you follow this pedigree, you can see that her male hemophiliac children or grandchildren often died young, right? So Leopold actually lived to 31, but we have a child here died at 3 and 11 and 20, and you can see it's all the males. Okay, another twist. What if neither allele dominates the other? All right, so first I said that you got a good allele and a bad allele, a dominant allele and a recessive allele. I said that the good gene could be dominant or recessive, right? But now I'm going to introduce something that's called incomplete dominance. And incomplete dominance and codominance are two different ways where we don't really see a recessive and dominant interaction. All right, so incomplete dominance. This is something in humans that would be the curliness of your hair, but we often show it in this picture here. We have, with this plant, a red flower and a white flower. Instead of all being red or all being white based on which allele is dominant, the F1 generation, you get pink, right? And then when you self-fertilize the F generation, you can see you get a couple of pink, the white shows back up again, and the red shows back up again. So this is a blending of the pigments that make up that petal, and we call this incomplete dominance. Codominance. An example of this is your blood type. And what your blood type is if we look on here, this is your red blood cells. Your red blood cells have a protein sticking off of it, but on that protein also can be sugars. So this is a big name here, right? But all I want you to remember is that there's a sugar on it. So the protein in purple shows up on all the different types of blood, whether you're O, A, B, or AB. The way we distinguish between whether you're one of those types is to look at the sugar. So what sugar's on there, we're going to call this the A sugar. This is the B sugar, and this is a combination of both A and B sugars. So O type have none at all. This is codominance because when in this individual here, they got an A sugar gene from their mom and a B sugar gene from their dad, nobody won out in that battle. Each red blood cell expresses and shows both of them. Okay, so your blood type is an example of codominance, where I'm going to see both of those gene products, both of those proteins or sugars in this case. And if we go back here to incomplete dominance, we see a whole different phenotype in incomplete dominance. I know that we weren't able to go through every one of the slides, but please go through those so that you can have a better understanding. I will have one last video to wrap up a few other terms related to genetics. Thank you.